Hi, today I am going to present the overall pipeline for all kinds of NGS data in just one lecture. So this will include the details, introductions about all kinds of NGS data as I have explained about RNA-seq data analysis in the previous lecture. So here it will cover the analysis of whole genome sequencing or whole exome sequencing, ChIP-seq data, ATAC-seq data analysis, which covers most of the applications of NGS in biological research as well as clinical and diagnostic research. So first, let's start from whole genome sequencing. So whole genome sequencing is basically meant for the analysis of all the gene expression data analysis, all kinds of mutational data analysis, including coding and non-coding reasons. So this is very important, but it is quite expensive in terms of NGS data generation. So this is basically a comprehensive method for analyzing the entire genome for a specific organism, and it provides the detailed information about genetic variations, mutations, structural variants or structural changes. So this is the brief summary of whole genome sequence. So for example, if someone wants to go for a basic analysis, let's say family-based whole genome sequencing, then two or more affected individuals, affected and unaffected individuals, then one affected individual. So exclude variants that are not shared by affected individuals that are present in unaffected individuals. Then exclude non-coding variants and coding variants that are found in the databases, for example, TCGA or 100 Genome Project, or that are not rare. So, for example, rare means greater than equal to 0.5%. Then test for segregation of identified genetic variants with disease-associated phenotypes. Sequence variants in ethnically matched controls. So this is kind of a basic goal if somebody is looking for. The same could be implemented for diagnostics or some other kind of targeted analysis. And the basic map or workflow could be, for example, sample input if you are going for the sample preparation to the analysis, complete analysis, then library preparation, sequencing from any company where you are interested or which are cost effective. Then this sequencing company will deliver the raw reads, for example, in FASTQ formats. And this FASTQ format will be processed by using Botai or some other softwares for read mapping, then BAM creation. So here you will get SAM file, then you convert the BAM file to SAM file, which was explained in the previous slides or previous lectures. Then go for BAM indexing, which is, let's say, sorting of the BAM files. And now you can go for analysis, for example, variant calling, copy number variation, then structural variants analysis, and some people could be who are interested in lower organisms which are not studied properly yet. They could jump for de novo sequencing or de novo analysis. Then go for final analysis, annotation of the filtered variants, then confirm the novel variants. Okay, so here the softwares could be used here, but the best thing is go for cufflinks or Galaxy. the integrated software platforms for NGS data analysis. Now, if you go for further summarizing of the workflow, it is read, raw reads, then coverage calculation, alignment, depth distribution, go for SNP distribution, structural variance analysis, insertion deletion analysis, and then their annotations or functional effects. Then next part could be if you want to go for figure analysis in terms of figure presentation, then it could be network of the targeted genes with highest SNPs or whatever you are looking for. And then the heat map of the selected genes. For example, these selected genes could be coming from these networks, which are, let's say, these are the genes which are highly connected genes with, in terms of the network connectivity. This could be annotated or plotted with the heat maps of gene expression or mutation or fold changes, p-values. Many more things could be plotted in the form of heat map or the other alternative way of plotting, which you feel is more attractive and more informative. So next, you could go for, let's say, a small RNA sequence analysis. This is also very common these days to go for a small RNA sequencing or miRNA sequencing. There are more things. Why I am bringing here? Because for most of the NGS data analysis, there is slight differences in pipeline for the analysis purpose. Most of the steps remains the same, for example, in the beginning and in the end during analysis or visualization. So this is the basic goal for small RNAs, and the pipeline would look like this. So there are many classes of small RNAs, tRNAs, miRNAs, so, and then snRNAs, etc. Less than 200 nucleotides in depth, each of which carries out distinct biochemical roles in the cell communication or signaling. These roles may include messenger RNA, that is mRNA, and then RNA interference, silencing, degradation, among others. These RNAs could be captured and sequenced using a variety of methods to enrich the for various classes. Then, for example, here, if you are looking for a pictorial presentation of the pipeline or analysis steps, so this could be one of the best view for understanding how to analyze and how to proceed for such a small RNA analysis. So then another thing is ChIP-seq data analysis, which is meant for transcription factor binding site enrichment analysis. And these things I will describe in details. So for example, if you are looking for the RNA-seq analysis, the pipeline was something like this. If you are going for whole genome sequencing, then you could go for gene expression analysis instead of RNA-seq. Let's say use it here, WGS or WES. So this could give you two different things: SNP profiling, differential express gene analysis. Then you could go for more analysis, network level understanding, pathway enrichment analysis, sample-wise SNP profiling, gene-wise SNP profiling. Then finally go for comparative study. Now, if you look for much better and advanced way of analysis and graphical presentation, then you could think how to plot these things. And these things are quite easy to be presented. Most of these steps are or were described in the previous lectures. So there should be no